Good morning. I am R. Padmavati, working as an assistant professor at Meenakshindra Engineering College, Kodambakam, Chennai. I am doing a PhD under the guidance of Dr. B. Rajamanan, Professor and Head, Faculty of Engineering and Technology, Annamalai University, Chidambaram, under the external guidance of Dr. S. Gunashegaran, Tansy Award, Dean and R&D, St. Peter's Institute of Higher Education and Research, Chennai. My paper title is Efficacy of Adapalene 0.1% and Benzoyl Peroxide 2.5% gel for the treatment of Acne Vulgaris using FTIR-ATR spectroscopic technique. Introduction. Acne Vulgaris is a dermatological disease with estimated 650 million people are affected, more than 95% of boys and 85% of girls, and it occurs on the face, neck, forehead, and then chest and then back. And it occurs due to it is a chronic disease and occurs due to the inflammation of the hair follicles and as well as the sebum producing sebaceous glands. And then the propionemakin bacteria, which lives in the absence of oxygen. These propionium bacteria take the control of the sebaceous glands, and it's widespread on the area of the skin and where the sebaceous glands are densely packed. And these glands produce a large volume of the sebum that provide a lipid-rich anaerobic environment that is optimum for the P. acnes. Specifically, the sebum of the patients with acne contains the lipoperoxides resulting from the peroxidation of the lipid squalene. And as well as the human sebaceous glands do not achieve the complete synthesis of the cholesterol, and thus it liberates the pure squalene. This treatment targets 4 factors which are responsible for the acne that increase the sebum production, and then keratinization, bacterial colonization, and the inflammation. The topical retinoids are effective in treating the inflammatory and non-inflammatory lesions by preventing the comedones and reducing the existing comedones and targeting the inflammation. The actual biomolecular changes due to the acne are present in the blood. The human tissue in the hair, which look like blood, will also reflect the same biomolecular changes present in the blood. Therefore, the single scalp hair tissues are collected from the 20 male and female between the age group of 20 to 25, and the gel are applied on a daily basis for the 3 months course. And then the FTIR-ATR spectrum are recorded for both the pre and then post treatment. And then the biomarkers of the for the acne vulgaris samples with the specific peaks during the before treatment were analyzed, and they are compared with the healthy subjects. The absorption values of the homotherapy bands of the biomolecules present in the hair samples both pre and post treatment of the subjects are noted from the overlay spectra, and it was observed that the biomarkers are significantly different between the pre and post treatment of the hair samples. And then further, they are validated with the statistical analysis by applying the dependent t-test, and which indicated that the spectral variations are statistically significant, and in the efficacy of the adapalene and benzoyl peroxide is estimated. And these are the types of acne: whitehead, blackhead, papules, and puzzle cystic and nodules. And these papules, puzzle cystic nodules are inflammatory, whereas the whitehead and black are the non-inflammatory. Pathogenesis: the overgrowth of the skin cells, and then excess sebum production, bacteriophagous inflammation, keratinization. This occurs due to when the skin consists of the 5 layers, due to the overproduction of the keratinization, the keratins, they clog the pores on the skin, and the propionine bacteria which lives in the hair follicles, which produces more and more sebum, and it damages the hair follicles also, and then causes acne. Here, the excess of sebum is caused by the androgen, the male and female hormones, which secrete more and more sebum, and the squalene byproducts, which also causes the excess sebum. The bacterial colonization, the bacteria, propionine bacteria, which lives in the absence of absence of oxygen, it produces a protein called inflammasome, which activate the more and more inflammatory molecules called the cytokines, I-alpha-1, I-alpha-6, and I-alpha-8, which causes the acne and also the inflammation. The other factors of the acne are diet, environment, pollution, and genetics. Diet, by taking the excess oily foods and then sweets, ice creams, wafers, chips. and as well as the environmental pollution, the petrol producing car that causes that produces the nitric oxide, which react with the volatile hydrocarbon present in the air, and that react with the ozone and produce a polluted ozone that react with the squalene and byproducts and causes the acne, and genetic factors also cause acne. Materials and methods. The human scalp tissues collected from the both healthy and acne affected patients, and they are first cleaned with the stone and then with the distilled water, and then they are taken through the laminar air in order to remove the water content present in the hair follicles. They are preserved in the plastic covers, room temperature, and they are placed on the FTIR-ATR accessory on the diamond crystal, and using the pressure there, the press applied in order having active contact with the crystal, and the FTIR-ATR spectrum is recorded. This is the mechanism action of the ADP and PBO. The ADP first it inhibits the commodore formation through its ability to bind to retinoic acid receptors and modulate the cell differentiation, and it also possesses the direct anti-inflammatory activity. The BPO, when it's applied on the skin, it rapidly metabolizes benzoic acid, and then it produces a more and more nascent oxygen from the peroxide group, and it kills the bacteria and that cure the acne. There is a treatment of acne. The topical retinoids, Adapalene, tazotrinone, tretinoin, and as well the moderate acne, or the tetracycline, minocycline, erythromycin, and doxycycline are used. And then the results and conclusion discussion, the science give an opportunity to use the hair sample as an hair samples to diagnose any diseases, and the most important components of the hair are fibrous proteins, then lipids, glycogens. So the biomolecular changes present in the hair samples could be well examined using the molecular spectroscopic technique, the FTIR-ATR. So we collect the 20 human hair samples from the acne patients, affected patients, before medication, then named as a pre-treatment, and for the same persons, the benzoyl peroxide and adapalene is applied for the 3 months course on a daily basis. And after the medication, 3 months, the single scalpel tissues are collected, they're named as a post-treatment. And FTIR-ATR spectrum are recorded for both the pre and post-treatment. And we see some remarkable differences between the pre and post-treatment. And so the comparative study is made, and the absorption values about the specific vibration bands of the biomolecule present in the scalp tissues for both post and healthier subjects are noted. In order to quantify the spectral signature, intensity ratio parameters are calculated with the absorption values of these specific bands. This intensity ratio calculated in order to rectify the error occurred due to the different thicknesses in the hair samples, and as well as the pressure applied during the spectral recordings, and the sensitivity exhibited by the FTIR bands of the protein, lipids, and squalene due to the higher absorption of the diseased and healthy tissue samples. Therefore, these are the biomarkers that are responsible for the disease acne. And as well as the 4 intensity ratio parameters R1, R2, R3, R4 have been introduced and calculated for the pre and post treatment of the acne Vulgaris tissue samples, and the significance of the intensity ratios or results are estimated using the parity test statistical method for both the pre-post and treatment, and as well as the post and the healthy ones. And we got the results, the P less than 0.05 indicate that there is a significant difference between the means of the internal ratio parameters calculated for pre and post treatment, and P greater than 0.05 indicates that there is no difference between the post and the healthy treatment. And the discrimination between the pre and post treatment have been thus proved from the statistical method. And efficacy also calculated more than 75% of result. In conclusion, this FTIR-ATR is the most powerful, quick, simple, and faster technique, because within this little span of the time period, we recorded the FTIR spectrum for the hair samples. And from the statistical data, the low value P less than 0.05 indicate that there is a significant difference between the means of the internal ratio parameters calculated for pre and post treatment hair samples. And then the R1, R2, R3, R4 were used for discrimination between the pre and post treatment of the acne Vulgaris disease, and thus efficacy of the gel adapalene 0.1 and Benzoyl Peroxide 2.5% shows the artifact results. Thank you.